Hi, I'm Sean Mooney, professor at the University of Washington and Chief Research Information Officer of Medicine. Over the past 2 years, we have been creating an initiative in medical data science, and as part of that, we have created this seminar. Speakers will include world-leading scientists who will tell us about how data can impact human health and their healthcare. Please use the link below to register for our mailing list to hear about future events in this series. We hope you enjoy them.
I'm really excited, a little bit starstruck to introduce Dr. Jason Moore today. We're really lucky to have him, and I want to thank him for being generous with his time. He is currently the chair of the Department of Computational Biomedicine at Cedars-Sinai Medical Center in Los Angeles. And he recently came over from the University of Pennsylvania, where he was the Edward Rose Professor of Informatics and director of the Penn Institute for biomedical informatics. Prior to being at UPenn, he was at Dartmouth until 2015, where he was the director of the Institute for Quantitative Biomedical Sciences. And prior to that, he was at Vanderbilt, where he served as the director of the Advanced Computing Center for Research and Education. He is trained at the University of Michigan and has a PhD in human genetics and an MS in applied statistics from that institution. His work has been communicated in more than 600 peer-reviewed papers, book chapters, and editorials, and he leads an active research program focused on the development of artificial intelligence and machine learning algorithms for the analysis of complex biomedical data. As I said before, we're really excited to have Dr. Moore today, and I think we're really lucky to hear him speak. So I'm definitely going to be enjoying the next 45 minutes to an hour, and I hope you do as well. And with that, I will turn it over to Dr. Moore. Thanks again.
Great. Thank you so much for the invitation to speak. It's always a pleasure to visit the University of Washington, even if it's virtually, and I know a lot of people there. Actually, my daughter was just admitted as an undergrad at University of Washington. I don't know if she'll decide to go there, but if she does, that'll send me up to Seattle a little bit more often. And so I'm absolutely thrilled to be on the West Coast and to be in Los Angeles. And we're building a new department, hiring faculty, hiring postdocs. So if you're a graduate student or a postdoc looking for a position, feel free to reach out to me. Be happy to see if you might be a good fit for what we're building here at Cedars-Sinai. And certainly if you see a way we can work together and collaborate, I'd love to talk to you. So the talk I'm going to give today is on our automated machine learning methodology and some thoughts about how to apply this to the automated analysis of genetics data. So this is how I think about what we call genetic architecture, or the relationship between the genome and the phenome. And this was a slide that was actually put together, or a figure that was put together by my PhD mentor, Charlie Singh at the University of Michigan. And it's really meant to illustrate the complexity of the kind of common diseases that we study, such as coronary artery disease. And so here we have the human genome and all of the various biochemical and physiologic systems that all interact with one another in the context of environmental exposures. And it's really all of our genetic information, environmental information, and our biology that together determines our individual trajectory toward a complex clinical endpoint, such as stenosis of your coronary artery. And what we do in human genetics is we typically take a single genetic variant out of this complex picture and ask whether it associates with a particular phenotype. And that's what genome-wide associations studies do with millions of single nucleotide polymorphisms. But that approach really ignores the complexity that we know is there, the biological complexity. And this really stems this of complexity really stems from, my thinking of health as a complex adaptive system. And in this slide here, we have, if we think about human health, we have many components. So down here, the black dots are genes, environmental factors, other biomarkers, metabolites, could be microbes, anything that you can measure. And those things organize themselves into subsystems and into what are called control structures. You can think of control structures as like a biochemical pathway, for example. And biochemical pathways interact to form a system, and it's the output of that system that really determines our health. And the point of a complex system is that health is an emergent property of this complex system, and it's really driven by the many biomolecular and system level interactions. And again, it doesn't make a lot of sense from a human genetics point of view to take a single black dot out of this picture and ask whether it's predictive of human health. You really need to understand and model all of the interactions, both in time and space, if you want to give yourself the best ability to predict who's going to develop a common disease. And of course, we live in a big data world where we can measure a lot of these factors now. And this is really why we need data science, and in particular artificial intelligence, because we have a very complex problem driven by non-additive interactions, and we have big data. And so this makes the problem somewhat intractable. And so our best hope to make sense of it and to develop useful predictive models for things like precision health, precision medicine, is artificial intelligence. And I've been developing artificial intelligence and machine learning methods for the genetic analysis of common human diseases my entire career, going back to graduate school. Now, unfortunately, as many of you know, machine learning is really hard. And one of the difficulties is that every machine learning method looks at data in a different way. On the left side of this slide are 3 different data sets with an X and a Y variable. So you can think of these as measures such as maybe weight and age. And you might be wanting to predict, say, risk of cardiovascular disease. So maybe the red dots are cases, people with cardiovascular disease, and the blue dots are healthy controls. And so what machine learning is really doing is trying to find a mathematical function that ideally, given the information about the X and the Y variables, is trying to discriminate between people with and without disease. And you can see from the patterns, the blue and red patterns in these figures. I love this figure, by the way. You can find this online easily by Googling machine learning and visualization. I love this figure because it really nicely illustrates how different machine learning methods look at the data differently and how their performance differs. So for example, on this first data set, a decision tree, which is a very simple machine learning method, actually performs the best, while on the second data set, it is not one of the better performing algorithms. And so this is one of the real challenges of machine learning, is that you don't know when you start an analysis with a new data set what the optimal machine learning method is to use. What we typically do is we gravitate toward something like gradient boosting that people say is the most powerful method, or we gravitate toward things we know how to do or understand or that might be interpretable, or things that we have experience with and know how to use. But that may not always be the best method for a particular data set. And as an example of this is some results from a set of benchmark data that we put together called the Penn Machine Learning Benchmark Data Set, PMLB, which is freely available on open source on GitHub. And this is about 160 classification data sets, and we cleaned them up and standardized them for things like scikit-learn for Python, so they're really super easy to use. And so in this particular paper that was published in Biodata Mining, we did an exhaustive search with parameter tuning across a bunch of different commonly used machine learning methods across all these different benchmark data sets. And by the way, some of these data sets come from the UC Irvine repository, some of them come from Kaggle, some of them are data sets that we had access to, like these middle ones here are actually simulated data sets. So this is a diversity of different kinds of data sets, different sizes and shapes, and et cetera. But you can see that there's a lot of diversity in performance across these different data sets for these different methods. Some data sets, all the methods perform well on, and some of the data sets, only certain methods perform well on. And what we did was we actually asked the question, how many times did each method outperform all the other methods on these 160 plus data sets? And the point of this slide is to show that even some of the simplest methods, like the naive Bayes methods, sometimes performed better than all the other methods, and reinforcing the idea that gradient boosting, for example, which performs pretty well across these data sets in the top row, sometimes the simpler methods outperform gradient boosting. So anyway, it's choosing the right machine learning method is difficult. And so this makes building a machine learning pipeline difficult. And so if you want to build a comprehensive machine learning pipeline to do an analysis, the first thing you need to do is pick a method. And as I just showed you, it's difficult to know what the right method is to pick. So algorithm selection is difficult. Then you need to tune the parameters. Once you've selected a method, every method has many, many different parameter settings, and that can be challenging. And then you might need to ensemble or stack multiple machine learning methods, and that's been shown to perform better than, say, single methods. And so you have to choose what methods are going to go into your ensemble. So that adds another layer of complexity. Then you might need to transform the features into a different transformation, like you might want to do a polynomial transformation, or you might want to normalize the features, or you might want to binarize the features, or do something like a one-hot encoding. And so there are tons of different feature transformation methods, and some of those have lots of parameter settings. And then you might want to engineer the features. So for example, in human genetics, we think about dominant and recessive and additive effects. Each of those requires a different, can require a different combination of features depending on whether you're looking at interactions, like a dominant by dominant interaction. And you might use something like a principal components analysis to do a linear transformation of the features. And so again, lots of methods, lots of parameter settings. Then you might want to do feature selection. I've published a number of papers on the Relief F family of methods, which I like, but you might want to do a univariate feature selection, or you might want to use random forest to do an importance score based feature selection. Tons of different feature selection methods, or you might want to use expert knowledge. That's the EK here. You could use biological knowledge. And then, of course, is data cleaning. And there are tons of different methods for cleaning your data and making sure that it's unbiased, et cetera. And so building a machine learning pipeline from A to Z requires a lot of knowledge, a lot of expertise, a lot of decisions that need to be made. And this can be very daunting, even for experts in machine learning, let alone novice users or naive users who want to get into it, or people that are not informaticians or computer scientists or statisticians. And then even once you have a pipeline that you're very happy with, then deploying that and doing something useful with that pipeline requires a whole bunch of additional work. You have to interpret the model, you have to validate it, maybe with independent data. You have to be able to explain it. How does it work? Why does it work? Do we trust it? All of those kinds of things. You might want to deploy it to the cloud. You might want to put a GUI on it. You might want to study the user experience. You have to be able to communicate it in a publication or a scientific presentation. Is it fair? Is it introducing bias from the data? And then, of course, doing clinical decision support with a machine learning model introduces a whole bunch of other complexities, such as building something like this into a clinical workflow that a clinician would actually pay attention to and not bog them down. So that's the motivation for my research program right now, and has been for the last 7 years. How do we make machine learning easy and accessible? And if you go back to the 1990s, we saw an explosion of user-friendly statistics packages, which really put statistics in the hands of everybody. And that's what I want to do. I want to make machine learning as easy and accessible as running a t-test or an analysis of variance on your data. And as you'll see, there's absolutely no reason why we can't do this. And this has led to a brand new discipline within machine learning called automated machine learning. So I got into this automated machine learning business, like I said, about 7 years ago. In the summer of 2015, I had just moved to Penn and had this new postdoctoral student come to my lab, Randy Olson, who was just finishing a PhD in computer science at Michigan State University. And I met Randy at a computer science conference, an AI conference, and he was giving a talk on artificial life. And I sat in the audience and I thought, wow, this guy is really amazing. He gave a phenomenal talk. He was doing really interesting research. So I went and talked to him afterwards. I said, look, I'm hiring postdocs. Would you like to come and work on AI methods in the biomedical space? And we went back and forth for a few months, and I finally convinced him to come. He was actually headed to industry, and I convinced him to come do an academic postdoc. And so Randy came, and the first summer he was with me, we kicked around some ideas, and he came to me and he said, I have this idea for automatically building machine learning pipelines. And we talked it through, and I thought it was a phenomenal idea. And we did a lot of whiteboarding and kind of sketched out how it would work and what it would do. And he went off and spent 6 months coding it up, and we then published our first paper on it that next year. And so the method we came up with is called the tree-based pipeline optimization tool, or Teapot, TPOT for short. And this is the GitHub links here. And I'm going to circulate after the PDF of these slides. So if anybody wants them, I'll send them to Sean, and he can forward it to the right person to send to all of you. But Teapot's completely programmed in Python. It's all open source. It's on GitHub. You can do whatever you want with it. And so this is the first paper we published, and I'll go through how it works here in a minute. But we went to a computer science conference, an AI conference, and Randy presented this paper. He gave a talk, and it was actually selected for a Best Paper award, which was pretty cool. And the paper is very widely cited now. It's getting cited over 100 times per year. And as it turns out, we were one of the first in the world, and I'm not exaggerating, to build an automated machine learning tool, publish it, make it open source. In fact, as evidence of that, we were 2 years ahead of Google. Google didn't release their first automated machine learning package until 2018. So I'd like to think that we and a few others who were working in this space around the same time gave Google the idea of jumping into the automated machine learning game. And this is their very first book on automated machine learning. And Frank Hutter, who's the first editor on the book, was also one of the first to develop an automated machine learning method. And so here's chapter 8 is on our Teapot method. So we have the distinction of being one of the first 3 or 4 automated machine learning algorithms ever developed. And it's not very often you get to say you were 2 years ahead of Google. So maybe that's the only time in my career I'll get to say that. But it's very proud of that. But and Teapot has been popular as a data science library, a Python-based data science library. This is a set of data that was published on the KD Nuggets website. If you're familiar with that, this is one of the big, most popular data science websites, and they did a survey of the top Python libraries for data science, and the number of stars on the GitHub project is on the y-axis here, and you can see Teapot is one of the top ones. It's actually on par with SciPy, which is very popular, and SHAP, which is very popular for interpretation of machine learning. So Teapot's getting a lot of downloads, a lot of notices, and a lot of likes, a lot of stars on GitHub. And it's actually the most, of all the automated machine learning methods, those are the purple circles. Teapot has the most stars. So let me step through Teapot and how it works. These are the 4 components that I want to talk about: the machine learning code base, how we represent the pipelines in the computer, how we optimize it, and how we control for overfitting, which is a concern. So the good news is we didn't have to develop the machine learning code base. We used the scikit-learn library, which I'm sure many of you are familiar with, which is probably the most widely used open source Python-based machine learning library out there. It's very professionally developed, does pretty much anything you want it to do in the machine learning space except deep learning. And so we took this as our code base and built Teapot around scikit-learn. Now, the next thing we needed was a way to represent the pipelines in the computer, and we used expression trees. And so this is a type of data structure that you learn about when you take computer science classes. And here's an example expression tree for this mathematical equation here. And so here you have mathematical functions in the nodes and digits here in the terminals of the tree. And it's very easy to traverse and execute binary expression trees, and there are lots of good algorithms for doing this. So this was a very convenient data structure for us because this is how we represent machine learning pipelines. So here's an example machine learning pipeline where you have a support vector machine in the root node with some parameters here. You have a function called combine features with two different operators here. This one selects the k best features from all the features with a parameter k. And then this one's a standard scalar that is doing a feature transformation on the features. So this transforms all the features using this standard scalar. This selects a subset of the untransformed features. This combines them and then runs a support vector machine. So this would be an example of a pipeline that Teapot could come up with automatically, and again works using these binary expression trees. So the next thing we needed was an optimization algorithm, and I have a lot of experience in the genetic programming space, and Randy was an expert in genetic programming, and so we decided to use that, which inherently operates on binary expression trees. And so the way genetic programming works is you initialize a population of solutions. So essentially generating, random pipelines. So here are two randomly generated pipelines. This one has a random forest, has the root node with a logistic regression, a normalized feature transformer, a relief F feature selector, for example, and a PCA feature engineering algorithm. So this would be an example of a machine learning pipeline that Teapot could develop and execute and evaluate. And we typically generate, say, 1,000, 100 or 500 or 1,000 such pipelines to start to generate variability of pipelines. We evaluate them all using something like an ROC curve. So we do classification, we evaluate the true positive rate, the false positive rate, and this serves as a fitness for each pipeline. And then we select the better performing pipelines. And the key to genetic programming is generating variability. So everything I've described thus far is just standard generating pipelines, evaluating them, selecting the best ones. And then what the secret sauce of genetic programming is swapping, taking two good pipelines and swapping different components of them through a virtual crossover operator using genetic terminology here. But basically, you're just picking good pipelines and generating variability in the pipelines by swapping different components. So the idea is that some of these, that there are modular components here that are high performing, and by swapping them in and out of different pipelines, you can explore different combinations, thereby generating better pipelines. And then you can, so this is just showing the two pipelines with their swapped components. So here's a simple flow chart for genetic programming. You generate 1,000 random pipelines, you evaluate them all by whatever criteria, you have some stopping criteria, you select the top performing ones, either probabilistically or explicitly. You apply these variation operators to generate new pipelines, and then you evaluate and you iterate until you stop. And genetic programming is a really, really wonderful discovery engine. And I highly recommend taking a look at it. I've published many, many papers on genetic programming and really like how it works. And then, of course, we worry about overfitting with any machine learning exercise, but in particular with an automated approach like this that's exploring thousands and thousands of pipelines or even millions of pipelines. We worry about overfitting. So we do two things. We do standard cross-validation, which if you do machine learning, you're very familiar with, where you divide the data into training and testing and perhaps even validation pieces. So Teapot does this inherently and it's built in. And scikit-learn has lots of cross-validation operators that we use and build into this. So this is one thing we do, but if you do a lot of machine learning, you know that cross-validation is not perfect and still susceptible to some overfitting. But what I really like is Pareto optimization. And this works really well with genetic programming, where you're evaluating, thousands or millions of solutions. And so if you're not familiar with Pareto optimization, think of this, every circle here is a different machine learning pipeline, and we are trying to minimize the error. But what we do in Pareto is we add a second axis here. You can even add a third axis with another fitness objective, or quality metric. And in this case, we do complexity. So thinking that bigger, more complex pipelines are more likely to overfit the data. And so you can also try to minimize the complexity. So what you do is you generate a bunch of pipelines, and you find what are called the Pareto optimal models here. This is called the non-dominated set, or the Pareto optimal models, where each one of these models, there's no one other model that's better at both criteria. And so that gives you this, what's called the Pareto front. So we actually select from this Pareto front when I said we select the best models. So here, this selection piece here, instead of selecting the one with the highest classification accuracy or the minimum classification error, we're actually selecting the best pipelines to generate variability and from this Pareto optimal space. And so this is nice because it gives you here are some models that perform really well but are very complex. Here are some models that don't perform well but are simpler. And the sweet spot's probably here in the middle somewhere. So this is also called multi-objective optimization, which I'm a big fan of, because we as humans don't make big life decisions based on single factors. When you go buy a car, you don't pick a car just based on the make or model or the price or the gas mileage. You weigh all of those things, and it's a Pareto optimal decision that you're making because you have to balance all the factors. You don't get everything you want. You have to pick and choose. And I think we should be doing machine learning the same way. So we've done extensive simulation studies to validate Teapot. This is a recent one that we just did, actually, using a brand new benchmark data set that we created called the DiGen resource. DiGen stands for diverse and generative. So this is a set of 40 benchmark data sets, all simulated, that we developed, where you know exactly what the generative function is. And we used an AI algorithm, which I'm not going to get into, to basically find a mathematical function shown here in the inset of some data that simulates data, the relationship between the features here shown at the bottom, the X1, X6, and X5, and a discrete clinical endpoint, such that one machine learning method performs really well. This is an ROC curve. And another machine learning method, like the logistic regression shown in purple here, performs poorly, and maximizing the variability of all the other machine learning methods shown here. And so this generative function, with this generative function, you can generate data that maximizes the diversity of performance of different machine learning algorithms. And we generated 40 of these with different combinations of best and worst performing, et cetera. And so we think this makes a really nice set of benchmarks that doesn't exist in the machine learning community. So this is on our GitHub repository under DiGen, and we have a paper under review that's on archive describing how we generated these data sets and how the details behind it. And this is a figure from that paper showing the diversity of performance across different machine learning algorithms, and this is hyperparameter tuned across the 40 benchmarks. So we applied Teapot to these data sets. So here are a number of different methods. Here's their performance in the box plots for the area under the ROC curve across the 40 data sets. You can see the spread of performance for each of the different machine learning methods. And here, these last couple here are Teapot. Even the worst performing Teapot pipeline that genetic programming generated did pretty well compared to all the other methods. And here's the best Teapot pipeline, the optimized Teapot pipeline actually performed the best. And AutoSclearn is widely regarded as one of the most powerful automated machine learning methods, and Teapot's doing a little bit better than AutoSclearn. Maybe not statistically different, but a little bit better on these data sets, but both are performing similarly. So Teapot is automatically able to find the best performing machine learning method across all 40 data sets. So this was just another validation that it's working quite well. Okay, so one of the questions in the early days of Teapot that I would get a lot is, okay, well, that's great, but how does this scale to big data? And we know genetic programming is computationally intensive, which it is, and that's a downside of that particular method. So we published this paper about 2 years ago. This is work done by my postdoc, Trang Lee, who's now working as a data scientist at Bristol Myers Squibb. And we did two things in this paper. The first thing we did was create what's called a feature set selector. This is a new operator for Teapot, thinking that, okay, instead of giving Teapot all the features, like if you're doing a GWAS, you have like 1 million features, right? Or if you're doing an RNA-seq, you know, you have tens of thousands of features. So that's a lot to throw at a machine learning algorithm in a computationally intensive way. So what we do is we take the genome-wide data set and we split it into small pieces. So if you're doing an RNA-seq, you could subset genes by pathway, or if you're doing a GWAS, you could do it by functional genomic element or something like that. So you end up with all these small data sets, and then the feature set selector is an operator in Teapot that goes into the tree that with a parameter that points to a particular data set. So Teapot over time can learn to select particular small data sets that to provide good performance at a fraction of the computational cost, because you're analyzing a much smaller data set with fewer features. So we did that, and then we also introduced what's called what we call a template, where instead of letting Teapot create any pipeline of any size and shape, you give it a template and say, okay, I just want a feature selector and a classifier. That's all I want. And then Teapot will work within that constraint to identify a best pipeline. And that can speed it up if you're worried about computational complexity. Okay, so I'm going to give next a quick overview of an application. This is a paper we published a few months ago in the IEEE ACM Transactions on Computational Biology and Bioinformatics, which is this is one of the top bioinformatics journals in the computer science space. So this was an application to GWAS data for coronary artery disease. This is data that came from the UK Biobank, 19,000 cases of CAD, 321,000 healthy controls. And what we did was we downsampled the controls to create a balanced data set to make the machine learning a little easier. And we did that 50 times just to control random sampling. And then we split into training and 2 different test splits so that we could have a final holdout data sets to evaluate the Teapot pipelines to make sure they were generalizable. And the first thing we did here was introduce what I would say is an expert knowledge feature selection step before we did the machine learning. And this is a paper I participated in a couple years ago with my colleague Volkert Osselbergs, who's a cardiologist informatician at the University of Utrecht in the Netherlands, and his postdoc Vinicius Tragante. And what we did was we really did a deep dive into coronary artery disease risk loci that have been identified through GWAS. And we said, okay, which of these are likely to be the best drug targets? And we used a lot of different sources of information, including information about the druggability of, given the protein structure, et cetera, et cetera. And we came up with a list of druggable risk genes with druggable genes with protein products that are druggable with risk loci from GWAS. And so what we did was we took these genes here and used this as a biological filter before we did Teapot as a way to make the problem more tractable, but also as a way to make the end result more interpretable and perhaps more impactful if your end goal is to develop new drugs for coronary artery disease. I highlighted in red this one here, this particular gene is the one that came up over and over again in our analysis that I'm gonna talk about. Okay, so here's what we did. we selected these druggable genes that I showed on the last list, and we identified connected genes based on their biological connections using a knowledge base called HedioNet, which integrates more than 20 different sources of knowledge about genes and drugs and pathways and diseases and side effects and symptoms, et cetera, using a graph database. So this is a Neo4j based graph database integrating all these different sources of knowledge, and then you can make inferences about things like biological connections. So we plugged our genes into this resource, and that we found the genes that were likely to have some biological connections. So we expanded the gene set out into that list. And then what we did was we extracted SNPs from the gene body, the promoter, and the enhancers for each pair of genes. And that became the feature sets that we used for the feature set selector in Teapot. So this is just a way to biologically filter SNPs using functional biological information and creating these feature subsets that Teapot could then use. So I'm not going to go through all the results, but this is the main result that we found. This was the best performing Teapot pipeline. And again, this was generated automatically. And this pipeline performed well across multiple different splits of the data and had good properties, et cetera. And it included that gene that I highlighted, one of the drug development genes, and a biological connector, the PRC1 gene. And there were about 28 SNPs between these two genes that went in, that Teapot selected. Remember, it's selecting from all the different pairs and subsets. It selected this pair and those 28 SNPs. And here's the pipeline that Teapot came up with automatically. And by the way, it had a testing accuracy of about 0.55 and holdout data, which doesn't sound like a big number for for machine learning. However, in the genetic space, the predictability of genetic information is not that good. And if you were to take all the GWAS hits for coronary artery disease and plug it into a machine learning algorithm, you would probably get a testing accuracy of about 0.55. So these 28 SNPs are performing quite well compared to what you would expect with genetic information. So here's the pipeline. The first thing Teapot does is a percentile selector. So it's selecting a certain percentage of the SNPs. So it's reducing the overall list from 28 to a smaller number. This is just illustrative here. And then that's passed on to a variance threshold selector. So that's applying a second round based on the variance. You think of selecting based on allele frequency or genotype frequency, selecting another subset from that subset. Then it's doing a stacking estimator feature engineering. So this is a stochastic gradient descent classifier that takes this set of SNPs, develops a predictive model, takes the output of that model and develops a new engineered feature that it puts back in the data set. Okay, so it's doing a feature engineering step here. Then it's doing a recursive feature selector using the extra trees classifier from scikit-learn. So it's doing another round of feature selection, and then it does a final round of extra trees classifier to make the final prediction of CAD. So this was significant based on permutation testing. It generalized independent data. It was the best model we found. And the key point here is that no human would ever in a million years develop this pipeline from scratch, right? Nobody would think to combine all these different factors, all these different operators together to do a machine learning analysis, but Teapot did it automatically. And this performs much better than any single machine learning method does. And so this is the value of automated machine learning, is coming up with these machine learning pipelines that you would never develop yourself. Now you could, from a practical level, you might say, well, this looks pretty complex and I don't feel comfortable with this. That's fine. You wouldn't have to use or deploy a pipeline like this. And if you didn't feel comfortable, you could at least use it to give you some ideas, like, wow, why is it doing all this feature selection? Maybe there's something important there. Or why is it doing this feature engineering? Or why is it using the extra trees classifier? Maybe I should focus on extra trees classifier as my machine learning method. So even if you don't feel comfortable using the output of Teapot, you could you could use the pipeline to give you ideas about how to build your own machine learning models that you do feel comfortable with. And this is a SHAP analysis, doing some interpretation, looking at the contribution of each feature here. These are the SNP RS numbers to the classification of each correctly classified case in the data set. And the point of this slide is to say, and these are SNPs in the PRC1 gene, these 3 SNPs here. And there were several clusters where different SNPs had different impacts. So you can see this cluster here, cluster 2, this SNP was driving a lot of the predictive value of the model. While cluster 1, it was this first SNP, this 178 SNP here, shown here on the left. And so what this does is this interpretation reveals the heterogeneity of the data. In other words, the SNPs that are contributing information in the predictive model are different in different individuals and contributing differently. And this is something that GWAS would never pick up on. In fact, this is something that GWAS completely ignored. And this gene, PRC1, was never found by GWAS as a GWAS hit for coronary artery disease. And so not only does this gene provide predictive value for coronary artery disease and the SNPs in it, but there's heterogeneity among the patients. And that might be why GWAS never found this, because GWAS is ignoring that heterogeneity. If you're testing this SNP here, this 178 SNP, its signal is going to get washed out because it doesn't play a very important role at all in all the other cases. This is the value of machine learning when looking at data like this and really digging into the details and interpreting it, is that the machine learning can capture the heterogeneity in a way that, say, logistic regression can't. Okay, so the last thought I want to end you with is what we're working on now. And I have a new data scientist in the lab, Audrey Ghosh, and a postdoc, Phil Frieda, that are working on an automated quantitative, not trait, automated quantitative trait locus analysis, AutoQTL. And so what we want to do is build on our success with automated machine learning. And we've developed several other automated machine learning methods, and turn that into knowledge that we can use to develop automated methods for particular bioinformatics tasks that we have, like Qtl analysis, where you're looking at trying to identify SNPs that affect a quantitative trait like gene expression level. So here's a Qtl where the mean gene expression is higher for one genotype than the others, and typically use something like linear regression to measure that relationship. But there's nothing special about linear regression. You could use machine learning for this purpose and perhaps capture more interesting genetic effects, interactions between multiple SNPs. And so that was our motivation for creating an automated quantitative trait locus analysis method. And this is the basic idea. Again, we're going to use genetic programming and binary expression trees. And this is what our models are going to look like. And actually, we have a prototype of this just working right now. I saw some of the preliminary results this morning. So I'm pretty excited about that. And I'm sorry I don't have those results to show you, but we should have more results in the next few weeks. So we just have a prototype working. But basically, what you want to do, the motivation for this is thinking about, okay, as a geneticist, what are all the decisions that you have to make when doing a competent Qtl analysis? Well, first of all, you have to pick a method. Most people use linear regression, but there's nothing special about linear regression other than, it's a parametric method that's easy to understand. You could use penalized regression. You could use regression trees or regression neural networks. There's lots of regression-based machine learning tools out there that you could use. So first decision is, okay, you have to select a method. Then you might want to do some feature engineering to put multiple SNPs together. Like I said, you might want to code a dominant by dominant or dominant by recessive relationship, and you could encode the SNPs in that way and provide that information. Or you could use, mathematical operators. Then you might want to do some feature selection based on allele frequency or biological function. Like you might want to pick, if you're interested in Qtl analysis, it might make sense to pick SNPs that are only in promoters and enhancers, for example. So you could use functional genomics information. You might use chromatin looping or transcription factor binding to guide your selection of SNPs. These are decisions a geneticist or a genomicist would make, and we want to give the algorithm the ability to do that automatically. And then I talked about feature encoding, coding dominant, recessive, or additive effects. That's another decision that geneticists usually choose an additive encoding, but again, there's nothing special about an additive encoding. There are plenty of dominant, recessive effects out there. So these are the raw materials, and AutoQTL will have access to all of this and automatically piece together a pipeline for doing a QTL analysis. So I'm really excited about this project, and hopefully in another month or two, we'll be able to start looking at real data. And if you're interested in automated machine learning, the book I showed earlier is certainly a good place to start, but in the more bioinformatics space, Elisabetta Manducci and my postdoc, Joe Romano, published this paper just a few months ago on reviewing automated machine learning with an eye toward genetic and genomic analysis of complex traits, and this was published in Human Genetics. Okay, I'm going to stop there, and hopefully there's time for a few questions. I want to thank Wei-Shan Fu, who did a lot of the coding for Teapot, Trang Lees, the postdoc with the feature set selector paper that I mentioned. Randy, of course, invented the Teapot method and deserves a lot of credit for bringing that to my lab, which I'm very excited about. The DiGen evaluation with the DiGen benchmark data was done by my data scientist, Pedro Ribeiro. Just recently, he just finished that last few weeks. We're writing up a paper on that. The Teapot GWAS paper was done by a former data scientist in my lab, Elisabetta Manducci. And then I mentioned Phil and Autry, who are doing the AutoQTL project. These are my two grants that fund my automated machine learning work. The first one is focused on cardiovascular disease, and that's where the Teapot GWAS paper came from, and we'll fund the AutoQTL work as well. And then this is a new R01 that I just got funded. This is a nice R01 focused on automated machine learning for the analysis of multimodal data for Alzheimer's disease. And we're part of an AI consortium for Alzheimer's, developing AI methods. This is my email address. Feel free to email me. My GitHub with Teapot and DiGen and some of the other projects, and my webpage. So I'll stop there and ask questions. And before I do, this is where we're located in West Hollywood. These are the West Hollywood Hills here. This is called the Pacific Design Center. My new department's on the 5th floor of the green building here. And this is expansion space. Cedars-Sinai is calling this the North Campus. The main hospital is just down the road here, about a 8 or 10 minute walk. So we're really close to the main hospital. But this represents a major new expansion at Cedars-Sinai in research, in biobanking and single cell and lots of other areas. And so the new department is really built to help expand the research enterprise at Cedars-Sinai. So I'll stop there and happy to take any questions that you have.
That was an excellent talk. Thank you so much. You're going to listen to, everybody clapping on mute. Thank you again for presenting. So we do have questions that are accumulating in the chat, and I'll start. I have the first one because I put mine in at the beginning. I'd love to hear your thoughts, Jason, on implementation and how we implement in practice. I think that's something that we're super interested up here. It feels like the Wild West still. There's a lot of industry that's interested. There is healthcare organizations that have analytics teams or data scientists that are implementing methods or taking like Epic's App Orchard methods and implementing them. And then there are academic data scientists like folks in Emerge, the AI group that you mentioned, there are others. How do we, where are we headed? And what's your view on how we ensure both accuracy, effectiveness of the methods, but also, show that these methods improve over time?
Yeah, that's a great question, Sean, and there's a lot to say about that. First of all, I think we're really AI in medicine, especially implementing AI for making clinical decisions, is in its infancy, and we're We're really in the beginning stages. I think the few success stories we've seen, like the the deep learning models for diagnosing diabetic retinopathy, are some great early wins. But those are examples where there's really a clear-cut imaging pattern for an AI to detect. I think making clinical decisions about what drugs to give and what treatments to give, and how somebody's going to respond to surgery, those kinds of things is enormously complex. Look, IBM Watson failed and they sold it off, right? And there's a good reason for that, because it's damn hard to predict clinical endpoints using AI. And so I think it's in its infancy. I think we have a lot of issues to work through. We have to work through the fairness and bias issues. The data, the data that we have in our electronic health records is enormously biased, right? Black patients are treated differently than white patients when they're triaged in the emergency room. And that shows up in the data that we analyze if you're looking at ICU data, for example. So, so we have a lot of work to do in the bias and fairness aspect, and that's a really new area of machine learning and AI that we have a lot of work to do in. Trust and explainability is a big area that we're really not good at yet. How do you make sure a clinician can trust an AI algorithm, whether it's something Epic's giving them or something we as academics are giving them? That's a big one, right? How do you make sure that, I think clinicians should be able to look under the hood and know exactly what the AI algorithm's doing. How is it taking, which pieces of information is it taking? How is it combining them to produce an output? And is that trustworthy? And is it explainable? Can you explain what an AI algorithm is doing? And I'm really worried about commercial solutions, because they don't show you, they don't let you look under the hood. They don't show you what the models are doing. They just say, trust us. And some of the Epic models are not generalizing, right? There have been papers published looking at really evaluating the Epic models, and they're not always generalizable to every hospital. So I could go on, but we have a lot of work to do. And then there's all the decision support. How do you create an alert? When do you create an alert? How do you work that into a clinician's workflow? What does a clinician do with that? So I think it's in its infancy, and I have to tell you, Sean, one of the reasons I'm really excited to be here at Cedars-Sinai is because I'm embedded in a hospital, and this was part of the decision making. And for me to come to Cedars-Sinai was to really have an impact, to really get in where I could be immersed in a hospital and really think deeply and work with clinicians on deploying AI tools like I talked about today. And we're doing a lot of work in the EHR space as well. I talked about genetics today, but we're doing a lot of work with these automated tools for electronic health record data and clinical outcomes as well. So I'm excited to be at a hospital. I'm excited to take on these challenges head on, but they're significant challenges. We have a lot of work to do in the AI space, and nobody should be fooled for a minute that this is going to have a big impact quickly.
Great, thank you for the answer. Bhargav, do you have time to ask your question?
Yeah, sure, I can. I think Dr. Moore, you kind of segued into it a little bit, which is nice, but I was kind of curious about what the discussion was around explainability when you were developing Teapot. If it started, and you talked about like the Pareto optimality, and you showed that figure. But kind of initially, was the goal always just to come up with the most accurate ML pipeline possible using the most robust kind of methods, or was complexity and explainability kind of a concern even from the beginning? And maybe like a follow-up would be, you talked about like maybe if the end user is a clinician, how has reception been if you've been able to gauge that from kind of end users? Are they like comfortable with more complex models, or does Teapot do a good job of coming up with less complex solutions that are still optimal and still understandable? That's just kind of what was going through my head when you were talking about some of those things.
Yeah, that's a great question. Explainability is very important to me. I'm actually a co-author on a huge paper that's coming out probably later this year on explainability and AI. So it's something I think a lot about. But you mentioned Pareto, and I think that's really key, that this idea of multi-objective optimization. And, the machine learning community, I think, is so naive about clinical and biomedical problems, because accuracy is not the only thing that we we as scientists, we as biomedical researchers care about, right? It's just like buying a car. You don't care about the color, you don't care about just the price, you care about everything. And so why is it that when we build a machine learning model that we key on the accuracy of the model? You know, so one of the advantages of Teapot is it's built around this multi-objective optimization. And so as you can see from my talk, I'm really interested in how we use expert knowledge to guide machine learning algorithms. And I think that's so critically important. And you can do that with Pareto. So if feature selection is part of your pipeline, you could evaluate the accuracy, but you could also evaluate, say, the druggability. You could give every gene a druggability score based on its protein structure and its accessibility to small molecules and those kinds of things. And you could evaluate a pipeline based on how many druggable genes it has in it, because those are more interesting to you. I published an editorial about this idea of interestingness, which we completely ignore when we do machine learning. How interesting is the model to a cancer researcher or to a cardiologist or to a neuropsychiatrist? And you can build that into your machine learning with Pareto optimization. And so I think that's a way to get at the explainability, and using the druggable genes as inputs in our Teapot pipelines makes it more explainable, makes it more understandable, makes it more interpretable on the other end, and more actionable, right? Which is something we care about, because we know the results at the end are going to be, we could predict coronary artery disease as well as GWAS with druggable genes and SNPs in druggable genes. And so that's really important. I think that gets at the explainability. And we need more human input into machine learning. That's another, I think, big mistake, especially people coming from the computer science make with machine learning, is that they're just throwing a naive machine learning algorithm at a problem. Some of you may remember this paper that Google published with UCSF and a couple other places a couple years ago, where they took a massive electronic health record data set with like 1 million patients, and they threw all their deep learning horsepower at it. And at the end of the day, if you look in the supplemental materials, it didn't do any better than logistic regression at predicting. And I would say, you know, maybe there were nothing but linear additive patterns in there, and logistic regression is all you need. Or maybe Google did not bother to use all the expert knowledge we have about you know, the features and the phenotypes to build a more robust machine learning model that wasn't looking at all the data, but doing feature selection, et cetera. So I think we have a lot of work to do in the explainability area, and it's not just SHAP values or feature importance scores. That's certainly part of it, but we have to get biological knowledge and human interaction inside AI algorithms.
Thank you. Appreciate it.
Great, thank you. William Harefield has a question in chat. Do you want to jump in and ask?
Yeah, that was an incredibly valuable resource. Your slides were assembled with a lot of insight. Thank you so much. I think it was Quantum Magazine that I read. It seemed like a real worrywart, insignificant problem, but I'm curious. In this magazine, it was stated that in the future, if we try to solve all major problems through machine learning, because the data sets are often so large and requires so many GPUs running full throttle to get millions and millions of parameters, we're not going to have enough service in the world, let alone we're going to exhaust all the resources to generate that much electricity. And then Teapot is going through an additional 1,000 pipelines. What are your thoughts concerning this worry?
Well, I think it's a significant concern, and I'm glad you raised it. I think, you know, we do need to worry about green energy, and one of the things I would say about automated machine learning, we published a paper recently on one of our other automated machine learning methods, which I didn't have time to talk about, where we put it head to head with a deep learning algorithm. The deep learning algorithm some of you might be familiar with the MIMIC data set, which is an ICU data set of 60,000 patients out of Boston. And somebody had published a paper where they developed a deep learning method and applied it to predicting sepsis using longitudinal data from the MIMIC data set. And it took 24 hours for the deep learning algorithm to run. Our other automated machine learning algorithm, which is called PenAI, was able to do it in less than 10 minutes and perform equally well. So I think this is an advantage of automated machine learning and not necessarily a disadvantage, is that it can arrive at an answer that's as good as deep learning, much faster and much easier. And you don't always need deep learning for every problem. And in this particular case, we showed that you absolutely did not need a deep learning method. An off-the-shelf machine learning method did just fine, and it executed in a fraction of the time. And you just needed automated machine learning to find the right method, the right parameter settings. So yes, Teapot can be computationally intensive, but I would say some of the alternatives that are being used, like deep learning, are even more computationally intensive. But this is a big problem, and I think it's something, you know, that we all need to think about. Do we really need to run those extra cycles? Do we really need, you know, is the gain that we're getting like the Google experience? God knows how many trees Google cut down when they did their massive electronic health record analysis, and at the end of the day, it did no better than logistic regression, right? Which they should have done in, you know, in the first place as the baseline. I'm sure the reviewers asked them to put that in there, and they buried it in the supplement. This is, I think we all need to take a close look at what we're doing, and whether the reward that we get from the machine learning is you know, worth the expense in terms of energy.
Thank you.
Thanks very much. So JT Bowler just has a comment. Will you share the book you recommended for automated machine learning?
Yeah, I'll put it in the chat. By the way, I'm okay on time. I'm happy to hang out a little bit longer.
We officially go till 2:30, but usually it doesn't take that long.
Oh, good.
But we're happy to keep chatting as long as we have discussing, as long as we have questions.
Awesome.
So David Chu asks, can you comment on Watson in genetics? You mentioned Watson earlier, so maybe you could expand on IBM Watson and your thoughts on it.
Yeah, you know, Watson, I have another talk I give on automated machine learning where I dig into the Watson a little bit more. So I'm happy to talk about that. And by the way, I'm putting the link to the book. It's an open access book, so you can get the entire book on PDF, which is nice. So Watson, yeah, Watson's an interesting story. You know, I think what happened with IBM is that they tried to corner the market on AI in medicine. You know, they developed Watson, which was a real tour de force, right? I mean, you have to give IBM credit. They put together a big team. They built Watson. They got it working. And, you know, combined machine learning and natural language processing and, you know, knowledge engineering and information retrieval, all the things we talk about in biomedical informatics. You know, they put all that stuff together and got it all to work. And it's really an amazing story. And then it beat the Jeopardy champion, right, in what, 2010, so more than 10 years ago. And so IBM realized they had something hot. And so what did? This is their big mistake. They decided, let's go after the healthcare industry. And what a bunch of idiots. Had they gone after, you know, had they started in engineering, for example, or something else like meteorology, maybe they would have had more success. But they jumped into the healthcare game. They tried to squeeze out all the competition. They rolled it out too fast, and, you know, convinced MD Anderson and Sloan Kettering and some other places to pay tens of millions of dollars for Watson. MD Anderson paid $60 million for Watson. $60 million. That's a lot of money, even for a wealthy institution like MD Anderson. And at the end of the day, of course, it didn't work as well as healthcare professionals. It underperformed, it underdelivered. And, you know, MD Anderson pulled out after spending $60 million and decided they had had enough. And so I think the mistake they made is they just pushed it into the healthcare space too quickly. It probably needed about another 10 years to cook. They should have started with some easier problems, maybe from the engineering space. And they got burned. And as a result, in January of this year, if you don't know, IBM Watson put it up for sale, and it was bought by an investment firm or a holding company or something, and they're going to chop it into bits and sell it. You know, probably sell it to China. I bet China's going to buy those pieces and parts of it and put it to use. So yeah, IBM just made a huge strategic blunder. I mean, many of us probably could have told them, hey, don't mess around with the healthcare space because it's really darn hard, right? So I think there's a good lesson in there, and that's what I was saying earlier, that AI, we have a lot of work to do, and there are not going to be any quick fixes, quick answers, quick solutions in the AI space. It's going to take a lot of real work, and I think IBM tried to bypass that, that lengthy process.
IBM had a lot of smart people. Why didn't they retrench themselves?
Why did they what? Why did they retrench?
Why didn't they consider the things you're talking about? Why didn't they go back and start over the project? Take Watson for what it is and retrench.
I think they had lost huge amounts of money, hundreds of millions of dollars on Watson, and they were just cutting their losses. And they didn't see companies need a business model, right? To continue to sink that much money into a project, they need to see that there's going to be light at the end of the tunnel, that eventually they're going to turn a profit. And they did not turn a profit with Watson and couldn't see a path to get there. So I think to make it work, they would have had to invest hundreds of millions of dollars over the next 5 or 10 years to do the things that I was talking about, and probably a whole bunch of other things that I haven't thought of, to get it to work. And they just weren't willing to do that. You know how businesses make those kinds of decisions. It's all profit-based decisions. But yeah, it would have been great if Watson would have retrenched and rolled up their sleeves and said, all right, let's go back to the drawing board for the next 5 years. We'll go underground and work on this and then come back, when we're ready. That's what they should have done. It's a sad story, it really is, because IBM Watson really ended the AI winter in 2010 with the Jeopardy, beating the Jeopardy champion. And that was exciting. And that's partly why we've had so much enthusiasm in AI the last 10 years, and, in addition to some other success stories. But yeah, it's a fascinating story. There'll be a lot of books written about it in the coming years.
Great, thank you. Arjun Chakraborty has a question. Do you want to jump off mute and ask, Arjun? Or I can read
Sure. First of all, thanks a lot for the great talk, Dr. Moore. I really enjoyed the talk and learned a lot from it. So you mentioned HeteoNet. I actually worked on HeteoNet before coming to do my PhD, and the newest version, which is Spoke. I had a question about, you mentioned graphical convolutional networks. You mentioned graph databases like HeteoNet, and I was wondering how they may be integrated with machine learning models to learn new things about biological processes.
Yeah, that's a great question. And we've actually taken HeteoNet. By the way, HeteoNet was developed by a former student of mine, Dan Himmelstein, who worked with me as a high school student and then a college student while I was at Dartmouth. And then he went to work with Casey Green, if you know Casey, who was also one of my former students. And then Dan went to UCSF, where he did his PhD and developed HeteoNet. So we've taken HeteoNet and updated it, and actually my Alzheimer grant that I mentioned, that's what we're doing. We're creating an Alzheimer specific knowledge base based on HeteoNet, adding some new things, adding some, the new version that we've created is ontology based, so it's going to be even more powerful and tailored toward Alzheimer's. And I have a postdoc, Joe Romano, who actually is on the job market. He's a fantastic guy, and he's been using it, developing it and using it for computational toxicology with really good success in combining it with graph neural networks, actually, which you mentioned. And so I think there's a lot of really interesting work at the interface between ontology-based graph databases, knowledge bases, and machine learning, in particular automated machine learning. But that is aim one of my Alzheimer grant, is to build an Alzheimer-specific knowledge base, and aim two is then to adapt automated machine learning algorithms to automatically use that knowledge. And that's what I was saying earlier. I really think that's the missing piece. One of the big missing pieces in a lot of machine learning is access to the knowledge, because what you want is an automated, and our other automated method, PenAI, can learn from experience. It actually has a MongoDB database attached to it where it stores the results, and it gets better with time, which we've demonstrated, which is pretty cool. But what we want to do is have those systems not only learn from their own experience, but learn from the expert knowledge bases to do things like feature selection, or to, you know, to figure out, they can even adaptively figure out what the best axis is to put in the Pareto optimization, right? Is it drugability? Is it, you know, is it some basic science notion about how genes work, or, you know? So I think this is going to be the big focus of my work the next couple of years, is integrating ontology-based graph databases with automated machine learning and trying to get those two things to work seamlessly together. I think there's a lot of interesting work in that space. I don't think I haven't seen anybody in the automated machine learning space working on knowledge bases yet. So I think it's wide open, and HediaNet was a really remarkable resource when Dan generated it. I think there's a lot of interesting things we can do with that. So I'm really happy to hear, Arjun, that you worked on HediaNet and you know about it. And I think graph databases in general are very much underutilized in the biomedical informatics and bioinformatics space. I think we could do a lot more with them. And I don't know, my brain is wired much more toward the graph database side of things than relational databases are.
Thanks for that. And I was wondering if I could ask a follow-up question to that. So you mentioned that well, one of the challenges to move healthcare forward will be, I think, to make the clinician work with these machine learning models, so so that they synergize. So I was wondering what you think about some of the challenges of that. Like, for example, for neural networks, there's one class of thought that says that neural networks are a model that's notorious for being like non-explainable. But there's one class, I mean, not one class, but like one group of academicians who say that they may be explainable in a certain sense. So what's the picture going forward, and how do we sort of make machine learning and healthcare synergize together to move move forward in the next generation of medicine and AI?
Yeah, that's a great question, Arjun. So, you know, the way I think about AI in medicine is we want our AI to look at data and make decisions like humans do, or possibly to do it better or in a different way that humans are not capable of. But let's stick with as humans do. So if we think about it from that perspective, if you're a clinician and you have a problematic patient and you're not sure what to do, what do? You walk down the hall and you talk to your colleagues, right? You've got everybody else in your department to rely and say, hey, I've got this case, I'm not sure what to do, what do you think? And your colleagues will say, well, based on my experience, I had a similar patient, or I've done this or done that, and when I treated a patient like this is what happened. And they give you advice. And if they tell you something like, oh, you should give this drug to this patient, you can ask them why. Why do you think that, right? And they can tell you why. They can explain why. And then you can internalize the answer and weigh that as you make a decision about treating a particular. So we as humans ask each other questions. We collaborate. We can ask why. We can ask for explanations. And that's an important part of our what we do for decision making. Now look at AI. The big problem with AI is you can't ask the why question. AI gives you an answer, but you can't say why. Why do you think that? Why are you telling me this? Why do you think this is the best answer? And no AI that I know about can do that, or at least it's commonly used today. A neural network can't do that. And I actually wrote an editorial about this that was published, an opinion piece that was published in Scientific American last summer, that this is the one question that medical AI can't answer, is why. And so we have a lot of work to do in that space. And here's an important reason for that, because we as humans learn from each other. And what you'd like to be able to do is learn from your AI. So if your AI is recommending you something and you ask it why, and it tells you why, you can internalize that and learn from it, and thereby improve your own understanding and your own decision making. So I think there's a give and take there that we need with AI, where we're learning from the AI, we can ask the why question. And similarly, you'd like the AI to be able to ask why. You know, if I tell the AI, I don't like your answer, the AI could say why. and I could explain why, and they could learn from it and use that to develop a better model. So hopefully that answers your question, but I think there's a lot of work to do in this why space that we're currently not focused on at all.
Thank you. And thanks for the great and informative talk.
Yep, you're welcome.
So I think I've gotten to the end of the questions in chat. Does anybody want to jump off mute and ask a question? All right, I'll take that as a no. All right, so Jason, thank you so much again. This is really a fascinating and informative talk. I think we all found it really, really interesting and really appreciate you willing to jump on Zoom. It's unfortunate that we're not doing talks in person right now. Hopefully next year we'll invite you back when we can get you up here for a couple of days and see all the work you're doing down in LA, because we're really excited you're here on the West Coast now, and have joined us on this side. So with that, I will thank you again, and we'll make, for everybody that's on, we'll make the recording available shortly. So thank you so much.
All right, thanks, Sean. Thanks, everyone. Bye-bye.